Hi everyone, it's Miss Lopez. In this video, I'm going to talk about the phospholipid model that we were using in class as notes. So we're going to attach this page to the mega sheet. And on the mega sheet side, we are going to take up half of the mega sheet, writing down the different labels for each of these molecules, identifying what they are, talking about their structure, and talking about their function. So we are going to break down the components of the plasma membrane, starting with large sections or large classifications, mainly the macromolecules. We're going to talk about each of the macromolecules, and we're going to identify the specific components of the plasma membrane that belong to each category. We're going to start with the lipids. And so you can write down this information directly on the mega sheet itself. The first lipid we're going to talk about is phospholipids. And these are item A. Let's start by talking about the function of phospholipids. Phospholipids make up the bulk of the membrane. They make up most of what the membrane is. And it's the phospholipids that are responsible for semi-permeability. In other words, allowing only certain things inside the cell. So that is the function of the phospholipid. Now let's look at the structure. So A1, which I've colored it in blue, is known as the head of the phospholipid, and it is hydrophilic or polar in nature. The tails of the phospholipid, however, they are nonpolar or hydrophobic, and I'm coloring these in red. And so when the question arises as to why the membrane is semi-permeable, why it's only allowing certain things in, it's going to be this section right here, the hydrophobic tails. There's two layers of phospholipids. It's known as the bilayer, and it's this inner section, which I call the hydrophobic core, that is actually preventing substances from crossing. Only non-polar substances are going to be able to pass this section. If a polar substance tries to approach and go through the molecules, this hydrophobic core is going to push back against it and not allow it to pass. The next lipid we're going to talk about is cholesterol. And it's this molecule over here. So it's identified as E. And you'll notice that it is found tucked away inside the membrane. And the job of cholesterol is to make sure that the membrane has the right fluidity. In other words, it's not too much like a liquid, but it's also not too much like a solid. So that's its function. As far as its structure goes, you can see that it kind of looks like these are rings of carbon molecules. So every point is a carbon molecule, but the carbon molecules are fused together, meaning that they are side by side. This is another carbon molecule, carbon ring, but you'll notice there's a bond right here. Cholesterol doesn't have that bond. And so we say that cholesterol, aside from being a lipid, therefore being made of carbon, hydrogen, and oxygen in no specific ratio, is made up of fused carbon rings. Okay, so those are the lipids that make up the plasma membrane. And remember that the number one molecule in the plasma membrane is the phospholipid, and that is why the membrane is semi-permeable. Next, we're going to move on to the proteins of the plasma membrane. Starting with this molecule right here, which is labeled as J, this is what's known as a peripheral protein. So aside from being made of carbon, hydrogen, oxygen, nitrogen, when we classify proteins, we look at their function within the molecule of the plasma membrane. So peripheral proteins, their function is mostly to stay on one side of the membrane. In this capacity or in this position, they can act as anchors. Many of them can act as receptors or have many other functions. But structurally speaking, peripheral proteins only stay on one side. And you'll notice that no part of this peripheral protein is touching the hydrophobic core. The next type of protein is called a transmembrane protein. And it's called a transmembrane because you can see right here, it starts on one side of the plasma membrane. So let's say this is the inside of the cell. And it goes all the way across to the outside of the membrane. Many students will call these channel proteins. That's fine. That's just a specific type of transmembrane protein. Now there is one very specific, so these channels are specific. In other words, they will only allow certain molecules through. And there's one molecule you need to know by name, or there's one transmembrane protein you need to know by name, and it is responsible for allowing water to cross the membrane. And that transmembrane protein's name is aquaporin. So aquaporin is the name of the channel that only allows water to cross the membrane. The next type of channel we're going to talk about is located right here as letter F. And I'm going to break down F even further. So F is referring to the entire protein. And you'll notice that for F, part of the protein is touching here by the phospholipid heads and the interior aqueous cytoplasm. And part of F is touching this up here with the hydrophobic tails. So this is where protein folding becomes very important. You want to make sure that all the amino acids that are going to touch the hydrophobic tails are also hydrophobic or non-polar. But all the amino acids that are touching here in this area, because this is aqueous and it's touching the heads, all the amino acids in this area must be hydrophilic or polar. Overall, this protein is known as an integral protein. So functionally speaking, it's going to be kind of similar to peripheral proteins. They have many, many functions, and some common examples are anchors, or they serve as receptors. But in terms of their structure, I'm going to break this down a little bit. So the section that I've labeled F1 is the part of the protein that is polar. It's going to be hydrophilic because it is touching the hydrophilic heads. So I'm trying to show this with these dots. Remember that proteins are made of amino acids, and it's the R group that changes from one amino acid to the next. So those dots are meant to represent amino acids, and those amino acids have polar R groups. Whereas these amino acids, located in F2 section, those amino acids are going to have polar or non-polar R groups because they're going to interact with the hydrophobic tails around them. So that's it for the proteins. Now we move on to the carbohydrates. And the first carbohydrate we are going to see, you'll notice, is located right here. So this carbohydrate is a special type of molecule that is known as a glycoprotein. So it's called a glycoprotein because this part up here with D, that is the carbohydrate, but the carbohydrate is attached to a protein molecule. And remember, we talked about these integral proteins. So this is an integral protein, can act as an anchor. So this carbohydrate is anchored on to this protein. So glycoproteins have many functions. Some of them are going to help for the binding of an egg and sperm. Some of these are specific types of molecules called mucins, and this is typically what you think of when you think of mucus. And the glycoproteins are there to protect very sensitive surfaces that are known as epithelial surfaces. So this would be like in your lungs, in your urinary tract, in the digestive tract, and reproductive tracts. So those are those combination of carbohydrate and proteins are kind of acting together. You can also have some that are involved in immunity. There's just a lot of different functions for glycoproteins. So there's your protein, and remember the dots represent R groups, and the red dots are hydrophobic or nonpolar R groups, whereas the blue dots are polar or hydrophilic R groups. And then for part D, that is the carbohydrate chain. So together, these form a glycoprotein. So the last carbohydrate we are going to talk about is this one right here. So here's that carbohydrate chain. And you'll notice that now the carbohydrate chain is attached to a lipid. This is what's known as a glycolipid. So this portion right here is the carbohydrate chain attached to the lipid. And the most important thing I need you to remember about these glycolipids is that they are involved with cell-to-cell interactions. The most common example is going to be blood typing. So if you're type A blood, your carbohydrate has a specific shape. If you're type B blood, your carbohydrate chain has a different shape. And so each of these carbohydrate chains have their different shape, and that's how we identify A versus B blood. If you have O-type blood, you're missing the carbohydrate chains completely. All right, and that is the components of the plasma membrane. So keep in mind that this plasma membrane, what they're showing you here, H through I, is a bilayer, having 2 layers of phospholipids. And for each of these components, you need to be able to identify each of the components of the plasma membrane. Describe the structure of each component, adding to what we already had learned in Unit 1 about each of these macromolecules, and describe the function of each component of the plasma membrane. I hope this video was helpful to y'all. Thanks for watching. Bye.